Welcome back to Medinare. In this video, let's discuss about clinical manifestations of Staphylococcus aureus in detail. Staphylococcus aureus is a pluripotent pathogen and it can cause diseases through different mechanisms which are toxin mediated and non-toxin mediated. It is responsible for causing both nosocomial infections, which are hospital-acquired infections, and community-based infections. It can range from minor skin infections to life-threatening systemic infections. So let's see what are those. So here are the list of infections caused by Staphylococcus aureus. It can cause skin and soft tissue infections, musculoskeletal infections, respiratory tract infections, bacteremia and its complications, urinary tract infections, toxin-mediated illness, and infections associated with community-associated methicillin-resistant Staphylococcus aureus. Now let's see what are the skin and soft tissue infections that this bacteria can cause. It may seem a lot, but trust me, I have some easy mnemonics for you to remember. The mnemonic for the skin and soft tissue infections caused by this bacteria is first found CM is hurry buying car. So let's see what are the infections. Folliculitis. Folliculitis refers to infection of hair follicles. Furuncle. Furuncle refers to boil and it manifests as painful pustular lesions in moist areas due to infection in the hair follicle. Carbuncle. These are severe painful lesions in the lower neck region extending to deeper subcutaneous tissue. It also manifests with mastitis and breast abscess. Impetigo also occurs in children. Usually it appears as red sores on the face that eventually bursts and it develops into honey-colored crust. Then we can also find some surgical site wound infection caused by this bacteria. Along with this, there is hydradentitis suppurativa, which is recurrent follicular infection seen in areas which are rich in apocrine glands, such as axilla. Then we have botryomycosis, which is a mycetoma-like condition, which is characterized by subcutaneous swelling, sinuses, and discharge containing granules. Also, cellulitis is seen as a manifestation of this bacteria. Moving on to the musculoskeletal infections. The mnemonic for the musculoskeletal infections is SOAP, S-O-A-P. Septic arthritis, osteomyelitis, pyomyositis, and abscess can happen as a result of musculoskeletal infection caused by this bacteria. Then we'll move on to the respiratory tract infections. The mnemonic for this respiratory tract infection is PEEPS, P-E-E-P-S, which is pneumonia, emphysema and pneumothorax, post-viral pneumonia, nematocele, and septic pulmonary emboli. Next, we have bacteremia and its complications. Bacteremia is because of the presence of the bacteria in the blood. And what can happen because of that? Sepsis, central line associated bloodstream infections, metastatic foci of infections, and infective endocarditis. The toxin-mediated illness caused by Staphylococcus aureus is staphylococcal scaled skin syndrome, toxic shock syndrome, and staphylococcal foot poisoning. We previously discussed about epidermolytic toxin or exfoliative toxin, which is one of the virulent factors which causes these infections, right? So this epidermolytic toxin is responsible for causing staphylococcal scaled skin syndrome. This syndrome can occur in newborn and children more often, and it may manifest with blisters and bullae formation. Also, Nikolsky sign is positive, which means we can see separation of the outer epidermal layer, leaving denuded underlying skin. The mucous membrane are usually spared, and this syndrome in newborn in its severe stage is called Ritter syndrome, which is characterized by fever, lethargy, and irritability with poor feeding. The milder forms would be pemphigus neonatum and bullous impetigo. Toxic shock syndrome occurs due to toxic shock syndrome toxin, which is also one of the virulent factors. It may manifest with fever, hypotension, mucosal hyperemia, vomiting, diarrhea, confusion, myalgia, and abdominal pain, and erythematous rashes. Then we have staphylococcal foot poisoning, which is because of the enterotoxin, which again belongs to one of those virulent factors. which we previously discussed. So, enterotoxin is expressed by nearly 50% of Staphylococcus aureus strains and is responsible for causing this food poisoning. It is a preformed toxin, and the site of action is vagus nerve and the vomiting center of the brain. All the symptoms like nausea, vomiting are common, and occasionally diarrhea and hypotension and dehydration can happen. Then, the infections associated with community associated methicillin resistant Staphylococcus aureus are necrotizing pneumonia and necrotizing fasciitis. Now, let's have a quick look at the laboratory diagnosis of Staphylococcus aureus. Initially, we need some sample to detect the bacteria in our body. So, depending on the type of infection, we need to collect different types of specimens. In case of suppurative lesions, the specimen collected are pus and wound swab. In respiratory infections, it is sputum. In urinary tract infection, it is midstream urine. In bacteremia, it is blood. In food poisoning, it is fecus, vomitus, and the food that caused that food poisoning. And carriers, nasal and perianal swab. After collecting these samples, firstly, what we do is direct smear microscopy. Gram staining of the pus or the wound swab would reveal pus cells with gram-positive cocci in clusters when seen under microscope. Next, we have certain cultures. Its detailed procedures will be explained in further videos. But for now, let's see the results. In nutrient agar, if the specimen is inoculated, it would produce golden yellow non-diffusible pigments made up of beta-carotene. In nutrient agar slope, it produces confident growth and looks like oil paint appearance. In blood agar, it is surrounded by a narrow zone of beta hemolysis. In McConkey agar, it produces small pink colonies due to lactose fermentation. And in liquid medium like peptone water, it produces uniform turbidity. And in selective media like mannitol salt agar, it produces yellow colored colonies due to mannitol fermentation. Next, we have some biochemical tests for identification. It is catalase test positive, heat stable thermonuclease test positive, DNase test positive, and phosphatase test positive. Mannitol sugar is fermented, and on potassium tellurate agar, it produces black colored colonies, and gelatin liquefaction is positive. Typing of Staphylococcus aureus can also be done to subspecies level for epidemiological purpose to trace the source of infection. It is used in case of outbreaks such as food poisoning in a larger community, when it affects a larger community. The types of typing methods are phenotypic methods, which includes bacteriophage typing and antibiogram typing, and genotypic methods such as PCR RFLP, which is restricted fragment length polymorphism, ribotyping, PFGE, and sequence-based typing. And that brings us to the end of the video. I hope you guys found it helpful. Do like this video and subscribe to Medinare for more. Thanks for watching.